Assalamu alaikum students, and today we will learn about the biological membrane, its structure and function. A living cell is a self-reproducing system of a molecules held inside a container. Every cell on earth uses such a membrane to separate and protect its chemical components from the outside environment through plasma membrane. Without membranes, there would be no cell and thus no life. The structure of plasma membrane is very simple. It serves as a barrier to prevent contents of the cell from escaping and mixing with the surrounding media. The plasma membrane does much more than that. We will discuss in detail in our coming slide. As shown in this figure, the simplest bacteria have only a single membrane, the plasma membrane, whereas in eukaryotic cells contain internal membranes that enclose intracellular compartments. These internal membranes form various organelles, including the endoplasmic reticulum, Golgi apparatus, and mitochondria. If a cell is to survive and grow, nutrients must pass inward across the plasma membrane and waste products must pass out. To facilitate this exchange, the membrane is penetrated by highly selective channels and transporters. These transporter proteins that allow specific small molecules and ions to be imported and exported. Other proteins in the membrane act as a sensor or receptor. We will discuss it in detail that enables the cell to receive information about the changes in its environment and respond to them in appropriate ways. The mechanical properties of the plasma membrane are equally remarkable. The plasma membrane is involved in cell communication, import and export of molecule, and cell growth and motility. If you look at the figure, receptor proteins in the plasma membrane enable the cell to receive signals from the environment. The transport protein in the membrane enable the import and export of small molecule. The flexibility of the membrane and its capacity for expansion allow the cell to grow, change shape and move. If the membrane is pierced, it neither collapses like a balloon nor remain torn. Instead, it quickly reseals. Okay, so let's discuss fluid mosaic model. Before 1970, cell biologists knew their cell membrane consists primarily of proteins and a double layer of lipid. But they did not know how these molecules were arranged within the membrane. In 1972, S. J. Singer and G. L. Nicholson proposed the fluid mosaic model of the cell membrane. A fluid is any substance whose molecule can flow past one another. A fluid includes gases, liquid, and cell membranes, which form the base for our understanding of the membrane structure and function. According to the fluid mosaic model, the cell membrane consists of a fluid formed by the bilayer of phospholipids with a variety of different embedded proteins forming a sort of mosaic, which means a patchwork within this fluid. A phospholipid consists of two very different parts. A head that is polar, so what is polar molecule where one area has a positive charge while another area has a negative charge. You can see in the lower picture. It consists of phosphate group and glycerol and hydrophilic in nature, means attracted toward water, and a pair of fatty acid tails that are non-polar and hydrophobic in nature. Cell membranes contain a variety of phospholipids with structures similar to those shown in the figure. Most phospholipids help isolate the cell from its surrounding, but some have other functions such as identification of cells. For example, lipid with carbohydrates attached on a red blood cells membrane and identify blood as type A, B, AB or O. Other glycolipids help in identifying a cell belonging to a specific individual. The components of cell membrane are very constant motion. To understand its mechanism, it is important to be aware that at any temperature about absolute zero, for example minus 273 degrees centigrade, atom, molecules and ion are in constant random motion. As the temperature increases, their rate of motion increases. At temperature that support life, these particles move rapidly. The phospholipid molecules in a membrane are not chemically bonded, so at a body temperature, their constant random motion causes them to shift about freely. The flexibility of the bilayer is very important for membrane function. If plasma membrane were stiff, your cell would break upon and die as you moved about. The fluid nature of membrane also allows membrane to merge with one another. To maintain flexibility, most membranes have a consistency similar to room temperature. For example, olive oil. But olive oil becomes a solid if you refrigerate it. All animals' cell membrane contain cholesterol, which is especially abundant in the plasma membrane. Interactions between cholesterol and phospholipid help to stabilize membrane, making it less fluid at higher temperatures and less solid at lower temperature. A high cholesterol content reduces the permeability of the membrane to a hydrophilic substance and small molecule that would otherwise diffuse through it. Reducing permeability allows the cell to exert greater control over which substance enter and leave it. Some biological molecules, including fat-soluble vitamins and steroid hormones, are hydrophobic and can diffuse directly through the phospholipid bilayer. However, most molecules used by a cell, including salt and sugars, are hydrophilic. Because these molecules are polar and water soluble, they cannot move through the nonpolar hydrophobic fatty acid tails of phospholipid bilayer. Movement of these substances in or out of the cells rely on the mosaic of proteins within the membrane. Remember, thousands of different membranes proteins are embedded within or attached to the surface of the phospholipid bilayer cell membrane. Many membrane proteins bear carbohydrate groups that project from the outer membrane surface. These proteins are called glycoproteins. Membrane proteins may be grouped into 5 major categories based on their function. These are enzymes, recognition protein, receptor protein, transport protein, and connection protein. So let's discuss in detail. Cells employ highly specific biological catalysts called enzymes, nearly all of which are protein. A variety of enzymes are associated with cell membranes performing different functions in the plasma membrane and in various organelles. For example, enzymes involved in ATP synthesis are embedded in the inner mitochondrial membrane. Plasma membrane enzymes help synthesize a supportive extracellular matrix that fills spaces between the animal cells. In cells lining the small intestine, plasma membrane enzymes complete the breakdown of carbohydrates and protein as these nutrients are taken into the cells. Next is recognition proteins, which are actually glycoproteins that serve as an identification tags. The cells of each individual organism bear distinctive glycoproteins that identify the cell as self. Okay, immune cells ignore self cells and attack invading cells or foreign cells, such as bacteria that have different recognition proteins on their membranes. For example, organ transplant, the most important recognition glycoproteins of the donor must match those of the recipient so the organ wouldn't be attacked by the recipient immune system. Transport proteins. Transport proteins spin the phospholipid bilayer and regulate the movement of hydrophilic molecules across the membrane. Some transport proteins form pores, mean ion channels that can be opened or closed to allow specific molecule to pass across the membrane. Other transport proteins bind substance and conduct them through the membrane, sometimes using cellular energy. Most cells bear dozens of types of receptor proteins, some of which are glycoproteins, that spin their plasma membrane. Each has a binding site specific for messenger molecules such as hormone or neurotransmitter, a nerve cell messenger. Okay, when the appropriate messenger molecule binds, it activates the receptor protein. Some membrane proteins act as both receptor and ion channels. One of the ways they do this is through direct receptor action. When a messenger molecule binds the receptor, it immediately and directly causes an ion channel within the same protein to open. The best known example of such a direct receptor action occur in a protein in the membranes of skeletal muscle cells, which help in the movement of body. This protein binds the neurotransmitter acetylcholine, which open an ion channels in the same protein and allows ion to flow that stimulate the muscle cell to contract. Other receptor proteins produce their effect through an indirect action. Upon binding a messenger molecule, which can be neurotransmitter or hormone, the receptor protein channels it shapes and biochemical activities. This starts a series of reactions within the cells that produce effects such as opening of many ion channels at different sites. Most neurotransmitters and hormones act in this manner. Connection proteins. A diverse group of connection proteins anchors cell membrane in various ways. Some connection proteins help maintain cell shape by linking the plasma membrane to the cell cytoskeleton. Other connection proteins spin the plasma membrane, linking the cytoskeleton inside the cell with the extracellular matrix outside, which helps anchor the cell in place within a tissue. Okay, let's review the cell membrane component of fluid mosaic model. As you all know that phospholipid consists of hydrophilic head and hydrophobic tail. Molecules with both hydrophilic and hydrophobic part are termed as amphipathic. It allows non-polar or non-charged molecules to pass, for example carbon dioxide and oxygen, that are freely passed through phospholipid bilayer. We also see these yellow structure which are called cholesterol. These are embedded within the tail of phospholipid bilayer. These are cholesterol molecules. It play a very important role. It helps to prevent phospholipid from separating too far from one another. Therefore, keeps the cell membrane compact and bound together and gives cell membrane flexibility. So cholesterol play very important role in overall health and function of plasma membrane. In the green chain of circles, these are carbohydrates. These carbohydrates are sometimes attached to the protein called glycoprotein and sometimes attached to the lipid called glycolipid. But these carbohydrates are sticking outside the membrane. They are used to identify and recognition purpose. For example, lipids with carbohydrate attached on the red blood cell membranes identify blood group as a type A, B, AB, or O. Other help in identifying a cell as belonging to a specific function. In the middle, you can see channels. These are actually protein channels. It is made up of protein, allows certain objects to pass, unlike a cell membrane which allow oxygen and carbon dioxide. But longer molecule or even charged molecule, for example glucose, is unable to pass through this lipid bilayer. But cell needs glucose, so glucose is actually taken in through these protein channels. Next is cytoskeleton. Cytoskeletons are actually thread-like structures that are formed in crisscross pattern. These are proteins and they are mostly microtubules and microfilaments, protein of cytoskeleton, which helps the internal support like frame of house. They also play a very important role for molecule transport. Molecules are moving along the protein thread of the cytoskeleton. So this is the reason why we call the plasma membrane as a fluid mosaic model. Because mosaic is actually it is made up of variety of parts. These parts can move around. The parts are not static. The phospholipid bilayer are able to move left and right the carbohydrates, the protein, the cholesterol molecules. So in this way, the viscosity of plasma membrane remains. So, two major properties of hydrocarbon tails affect how tightly they pack in the bilayer. First one is their length and the second is the number of double bonds they contain. A shorter chain length reduces the tendency of the hydrocarbon tails to interact with one another and therefore increase the viscosity of the bilayer. The hydrocarbon tails of the membrane phospholipids vary in length between 14 and 24 carbon atoms, with 18 to 20 atoms being the most usual. Most phospholipids contain one hydrocarbon tail that has one or more double bonds between adjacent carbon atom and a second tail with a single bond only. The chain that contains a double bond does not contain the maximum number of hydrogen atoms, so said to be unsaturated with respect to hydrogen. The hydrocarbon tail with no double bonds has a full complement of hydrogen atoms and is said to be saturated. Each double bond in an unsaturated tail creates a small kink in the tail, means they bend, which makes it more difficult for the tail to pack against one another. For this reason, lipid bilayers that contain a large proportion of the unsaturated hydrocarbon tails are more fluid than those with lower proportions. As the temperature falls, they incorporate phospholipid containing more unsaturated fatty acid into their membrane. When the temperature rises, the organism restores the saturated phospholipid. How fluid a membrane is at a given temperature is strongly influenced by relative amount of saturated and unsaturated fatty acid tails in a membrane phospholipid. Saturated fatty acid with no double bonded carbon are stray and can pack tightly together, forming a relatively stuffed membrane. Unsaturated fatty acids have one or more double bonded carbon, each introducing a kink into the tail, as you can see in the figure. This structure forces the phospholipids further apart, making the membrane more fluid. Okay, in animal cell, membrane viscosity is modulated by the inclusion of the sterol cholesterol. This molecule is present in especially large amount in the plasma membrane, where it makes nearly 20% of the lipids in the membrane by weight. Because cholesterol molecules are short and rigid, they fill the space between neighboring phospholipid molecules left by the kinks in their unsaturated hydrocarbon tails, as you can see in the picture. In this way, cholesterol tend to stiffen the bilayer, making it less flexible as well as less permeable. Okay student, this is all about plasma membrane. Hope you understood the lecture. If you have any question, we will discuss it in our question answer session. Thank you.